Hey people, Wadlinger here. Let's go over Electron Transport Chain. This is the grand finale of cellular respiration, and we want to make all the energy out of the electron carriers that we've been collecting so far. So, here we are in the mitochondria. You can see our outer membrane, our inner membrane, and our intermembrane space. Until now, we just did the citric acid cycle, which took place here in the mitochondrial matrix. So you can see we've got a couple of enzymes going for this electron transport chain. We've got NADH reductase at the start. That's going to interact with NADH. Then we've got two other transporters. And then at the very end, not really part of the electron transport chain, we have ATP synthase, which is going to make our ATP. So let's get going. We're going to start with NADH. Remember that NADH picked up our high energy electrons from our glucose molecule in all the phases we've had so far. And it's going to get oxidized back to NAD+, and it's going to drop off its two high energy electrons at NADH reductase. So we've now reduced this enzyme. And that enzyme is going to use that energy to do some active transport. It's going to take a pair of hydrogen ions and it's going to transport them up into the intermembrane space. And we're then going to pass these electrons on to the next transporter, which is going to use them to power more active transport. Notice we're transporting up into the intermembrane space where there's a higher concentration. We're going to pass these electrons on again to our last transporter. So those same two electrons that we got from NADH. And we're going to power more active transport. At this point, our electrons went from high energy to low energy. So it's time to get rid of them. We need the garbage collector. So in this case, garbage collector is oxygen. Oxygen's going to come in, breathe it in, and it's going to pick up those electrons, and it's going to pick up some hydrogen ions, and that will create water for us, which will then diffuse out of the mitochondria and out of the cell, and we'll breathe it out. There it goes. Okay? So you might ask yourself, why have we now created this concentration gradient? Well, now it's time to make the ATP. So we're going to use ATP synthase, and we're going to take those hydrogen ions and we're going to let them go back to equilibrium. As they do this, they're going to spin the enzyme like a revolving door, and it's going to take ADP and a phosphate and jam them together. So we'll end up with ADP plus a phosphate turning into ATP, which is exactly what we wanted. So for every pair of hydrogen ions that go through, we're going to make one ATP. If you look, we activated 1, 2, 3 of the protein pumps, the transporters. So we're going to make 1, 2, 3 ATP. Now you might ask yourself, what about FADH2? So FADH2 is not quite the same because FADH2 cannot attach to an enzyme NADH reductase. So what's going to look different here is that FADH2 is going to have to start at the second transporter. And that's going to turn it back into FAD, which can then be reused in the citric acid cycle. So because these two electrons are now only at the second transporter, they're going to power active transport, and then they'll get passed on, and they'll power active transport, and then oxygen will take them away with some hydrogen as water, just the same. But because we only powered 2 of our active transporters, we're only going to be able to make 2 ATP. Now, this last bit where ATP synthase turns ADP and phosphate into ATP using that concentration gradient happens to also go by another name. The other name for this is oxidative phosphorylation. This should make sense to you because it's all happened by doing a series of redox reactions. We've taken electrons and we've passed them from one molecule to the next in a series of redox reactions. So we've done phosphorylation, adding phosphate to ADP to make it ATP, through oxidation, through moving electrons around. Hope that helped.